A diverse group of biological macromolecules, the lipids, part 3 of chapter 3. Lipids are a diverse group of nonpolar hydrocarbons. When we take a look at long-chain hydrocarbons, we do realize that the carbons and the hydrogens generally tend to have no net charge, positive or negative. Because of that, we describe them as nonpolar, and nonpolar molecules will not orient themselves towards water. We call them hydrophobic. And so lipids are a great category of very diverse molecules that have that general characteristic of not mixing with water. If we take a look at the different types of categories of lipids, there are fats, oils, waxes like the beeswax, phospholipids, and steroids. We do know, and we have already mentioned, that the energy stored in the bonds of hydrocarbons are a great source of chemical energy for organisms. Not only can they be a storage source for energy, lipids can be sources of insulation and even protection from the environment, as well as hormones and other kinds of components that are used in the formation of cell structure. For fats and oils, the first general example that we give to students about lipids are the neutral fats. Neutral fats contain two main components, a 3-carbon alcohol, sugar alcohol called glycerol, and then 3 long-chain fatty acids. When we combine the two with the 3-carbon glycerol backbone and the 3 separate long-chain fatty acids, we form what's called a triacylglycerol, or a triglyceride. We are going to attach the fatty acids to the glycerol backbone by something that's called an ester linkage. Again, that's just a special name for the type of covalent linkage or bonding that's used to attach fatty acids to the glycerol backbone to make a single unit or molecule of a triglyceride. So here's an illustration of what we just described. At the top is glycerol. Notice it has the 3 hydroxyl functional groups as part of its structure. We're going to attach one separate fatty acid chain to each one of those hydroxyl groups. The fatty acid is listed or illustrated in the middle, and we call it a fatty acid because it has a carboxyl functional group on one end. And remember, every time we see a carboxyl functional group attached as part of the structure of a molecule, we automatically give that molecule the name of an acid, and then we have this long hydrocarbon chain. And that's, of course, where we see that stored chemical energy in the form of these single covalent bonds. Here at the bottom is the whole entire structure: the glycerol backbone, the ester linkage, and the long hydrocarbon chains attached to the glycerol backbone. 1, 2, and 3. For the fatty acids can come in 2 general forms, saturated and unsaturated. When we say a fatty acid is saturated, it means that every single carbon has a single covalent bond with a hydrogen attached, not including the carboxyl group. So we're talking about the hydrocarbon chain. That's part of the fundamental structure of a fatty acid. When we have saturated fats and fatty acids, we find that these can be packed together tightly because these are long linear chains. In contrast, unsaturated fatty acids have at least one double bond somewhere in the hydrocarbon chain. That one single double bond means that we don't have hydrogens attached to every carbon for the location of the double bond. We simply have a single hydrogen. That carbon is unsaturated. When we have these double bonds somewhere within the chain of the carbons and hydrogens, we're going to find that depending upon which side of the hydrocarbon chain the hydrogens that are used in the formation of the double bond, are they both on the same side or are they on opposite sides, is going to give this unsaturated fatty acid some unique structural characteristics. Generally, unsaturated fats that are composed from unsaturated fatty acids are liquid at room temperature. We call those oils. If it's a saturated fat, we're going to find that it's usually solid at room temperature. Things like butter, lard, Crisco, those are examples of, and even coconut oil, those are examples of saturated fats that are solid at room temperature. The reason that the unsaturated fats tend to be liquid or oils is because the location of the double bond can form a kink. If the hydrogens are in what we call the cis arrangement on the hydrocarbon chain, then we see a kink, and that keeps the molecules separated from one another because of that bend, and we see that that type of unsaturated oil or fatty acid, like oleic acid, is liquid at room temperature. If we have trans fats, and you've probably heard about trans fats, the reason it was discovered that those aren't so good for you is that trans fats have the hydrogens on opposite sides of the double bond, which means at room temperature they're more likely to be solid lipids or solid sources of fats. And we now know, based on research that's been done, that foods that are high in content of these trans fats, especially ones that are used in sort of fast food industry particularly, but any of those trans fats that are solid at room temperature tend to be less beneficial, much less beneficial for the human body than the lipids that are made with unsaturated fatty acids that are the cis form of fatty acid chains. Here we have an example of another fatty acid, but it's not really even giving us that idea that we have a linear arrangement because we have multiple double bonds. Notice that we have 1, 2, 3 double bonds that are found in the hydrocarbon chain. Because there are 3 double bonds, this hydrocarbon tail actually forms this sort of this little kink or this C shape. That's the type of structure we want to see in the most beneficial types of fatty acids. We call them omega-3s, omega-6, and this is an example of alpha-linoleic acid, which is an omega-3 fatty acid, which now, based on research, and of course sometimes our ancestors even knew, based on consumption, that these are some of the healthier fats and healthier sources of the very beneficial fatty acids, often found in fish, like salmon, trout, tuna, any of the oily, what are called oily fishes, usually have very high concentrations of these omega-3 fatty acids. Consumption of these omega fatty acids can reduce our risk of heart attack or heart disease, reduce our levels of triglycerides in our bloodstream, and can even be helpful in reducing blood pressure. Waxes are types of lipids that are, again, are made of long-chain fatty acids that are so dense in their construction that they're, just like the beeswax in a honeycomb, going to be hydrophobic or prevent water from soaking in. And so wax is an often helpful component on the surface of leaves, on the feathers of birds, and any type of organism on Earth that needs to protect itself from water, like rain or a watery environment, can use waxes in their construction to protect the surfaces of those organisms. Phospholipids we'll talk about in more detail in another chapter when we talk about biological membranes, but phospholipids are similar to neutral fats like triglycerides with one very significant difference. So if we take a look here in this illustration on the right, we have a glycerol backbone right here in the middle. This glycerol backbone has 2 long-chain fatty acids attached, but in the number 3 position, the number 3 carbon, instead of a 3rd fatty acid chain attached, we instead have what's called the phosphate group. And if you go back to our discussion about functional groups, I mentioned the phosphate group is a vitally important functional group, And here we see an example of how nature has utilized that functional group. The phosphate group is attached to the number 3 carbon on the glycerol backbone, and to that phosphate group is another side group. The side group can be very large, and oftentimes examples of side groups that are attached to these phosphate groups, when we look at phospholipids, can be things like choline and serine. So, if you take a look at the amino acid serine in our amino acid table in one of our other discussions, you can see how this side group can be pretty bulky, fairly large. So, phospholipid, this is the general sort of schematic and illustration that's used to describe or define a phospholipid. Well, so what's important about that? Well, this structure has some unique interactive capabilities simply due to the hydrophobic tails that are made of long-chain hydrocarbons, which are nonpolar, and this was often described as a head of the phospholipid, which is highly charged. If you take a look at this phosphate functional group, we have at least one full negative charge, and some other illustrations will show more than one full negative charge. The side group often has some type of negative charge to it, and so we describe the head of the phospholipid as polar and hydrophilic. So the head of the phospholipid is hydrophilic or water-loving. The tails of the phospholipid, the fatty acid chains, are hydrophobic or water-fearing, and this structure all by itself, if you place it in water, will naturally arrange itself with the polar head toward the water and the nonpolar tails away from the water. Here we have examples of other ways that we can depict the structure of a phospholipid. Either this larger illustration showing you a little bit more detail. Here's a space-filling model showing you the exact same thing with a little bit more realistic arrangement of what atoms and size of atoms are used. And then the little cartoon drawing, which is the one I learned about when I was taking my first biology and chemistry classes. And I remember when I first looked at these illustrations in the textbook, because they weren't very informative, I was trying to figure out what is this round thing and what are these long tails. So we now know that students need more information than this little cartoon drawing of a phospholipid. They need to know a little bit more about the different atoms of different elements that are used, and that's why the space-filling model and this more detailed cartoon drawing on the far left are better sources of information so that students can understand the structure of a phospholipid. And then when we further describe biological membranes, hopefully it's more evident or more understandable how these individual phospholipids can organize themselves without any extra energy or any extra help, if you will, to create those biological membranes. Here we have an example of the arrangement of phospholipids forming what's called a phospholipid bilayer. So 2 layers of phospholipids, the hydrophobic tails pointing toward one another, and the hydrophilic heads pointing away or toward the watery environment on either side. We have these structures when they have 2 characteristics like this with polar and nonpolar features. We call this an amphipathic molecule. It's got polar and nonpolar. It's got amphi means 2, so it's got 2 characteristics. Another category of lipids are the steroids. The steroids are made of ring structures, or these geometric shapes, and we have sort of a very fundamental template of a steroid, which I call 3 stop signs in a house, that are used in the construction of a variety of vitally important chemicals or lipids, especially in mammals. We call cholesterol, cortisol, and vitamin D. Notice we have a little short hydrocarbon tail on cholesterol, tiny little tail here on cortisol, but it's this steroid template that's fundamental in the structure of vitamin D, cortisol, and cholesterol. These are all going to be categorized as types of lipids. The steroids are going to have all of those same characteristics that we assign to a lipid of being generally hydrophobic, which means they're insoluble in water. And even some of our hormones that we rely on for our sex characteristics or for reproduction, things like testosterone, estradiol, those are also going to have a steroid hormone template in the structure of those biochemicals.